Welcome back to our channel. Today we are going to see new topic, Simple Microscope. A simple microscope is an optical instrument consisting of a single convex lens that produces a visual, erect and magnified image of an object placed within its focal length. History of the simple microscope. The simple microscope was developed during the 17th century. The Dutch scientist Antony van Leeuwenhoek made powerful single-lens microscopes capable of magnifying objects up to 200 to 300 times. Using these microscopes, he became the first person to observe microorganisms such as bacteria, protozoa, sperm cells, and blood cells. His discoveries laid the foundation for microbiology, and he is known as the father of microbiology. Principle of simple microscope. The simple microscope works on the principle of refraction of light. When an object is placed between the optical center and the principal focus of a convex lens, the light rays passing through the lens bend and form a visual, erect and enlarged image on the same side of the lens as the object. The magnification increases as the focal length of the lens decreases. Construction of a simple microscope. A simple microscope consists of the following parts. First, convex lens, the most important part of the microscope. It is made of high quality glass, responsible for magnifying the object. Second, lens frame, holds the convex lens finely, protects the lens from damage. Third, handle or stand, helps in holding or supporting the microscope, makes observation easier. Fourth, object holder. Some simple microscopes have a stage or holder to place the object. Fifth, light source. Natural sunlight or an artificial lamp provides illumination. Working of a simple microscope. First, place the object below the convex lens. Second, bring the lens close until the object lies within the focal length. Third, light from the object enters the convex lens. Fourth, the lens refracts the light rays. Fifth, a visual, erect and magnified image is formed. Sixth, the observer views the enlarged image through the lens. Magnification power. The magnifying power of a simple microscope depends upon the focal length of the convex lens. Formula for a relaxed eye. M is equal to D divided by F. Formula when the image is at the least distance of distinct vision. M is equal to 1 plus D divided by F. It is refers to M is magnifying power. D is equal to least distance of distinct vision. F is equal to focal length of the convex lens. Characteristics of the simple microscope. Uses only one convex lens. Produces a visual image. Image is erect and magnified. How low magnification compared to a compound microscope. Easy to use and maintain. Portable and inexpensive. Requires proper lighting for clear observation. Advantage of the simple microscope. Simple construction, low cost, lightweight and portable, easy to handle, no electricity required, bright image due to fewer lenses, useful for educational purpose, suitable for examining small objects quickly. Disadvantages of the simple microscope. Low magnification, low resolving power, cannot observe bacteria, viruses or cell organs clearly, narrow field of view, less suitable for scientific research. Applications of simple microscope. Simple microscopes in applied in biology, studying leaves, flowers, seeds and insects, observing plant and animal tissues. In medicine, primarily examining of skin, hair and nails. In jewelry, inspecting diamonds, gemstones, and gold ornaments. In watch repair, examining tiny gears and springs. In electrons, inspecting printed circuit boards and small electronic components. In geology, studying rocks, minerals, and fossils. In education, demonstrating microscopic observation in schools and colleges. Today's question. First, what is the magnifying power for a relaxed eye? Second, is the image inverted or erect? Thank you for watching the video. Share your thoughts in comment box about this video. If you found this content valuable, don't forget to like, share and subscribe for more updates. Thank you.